component called DNA that shape our bodily functions and its structure. Okay. So these are the learning objectives which I will cover during the presentation. It includes the definition of DNA. Then I will distinguish between DNA, gene and chromosome. Then you will be able to interpret the DNA double helix model, and you will also be able to design a DNA model. Okay. So my methodology will be inquiry based learning, and the assessment techniques will be MCQ. So the DNA is the terms full form is deoxyribonucleic acid. Okay. So DNA is basically a hereditary material that is present in our body, and it stores the genetic information that determines an organism characteristic and traits. So the function of DNA is it synthesizes the proteins. So these are the scientists who contributed in the discovery of DNA, starting from Frederick Fisher, who isolated the DNA in 1869. Then we have Erwin Chargaff, who gave the Chargaff rules in 1950. Then we have Rosalind Franklin, who by the X-ray diffraction she gave the structure of bDNA. Okay. Then we have Francis Crick and James Watson, who gave the 3D DNA structure in 1953. So we basically get confused between the chromosome, DNA and what? Our gene. So now let's understand what is the basic difference between these three terms. So chromosome and what? First let's see. This is the human. In this human we have billions of what? Cells. In the cells we have the nucleus. In the nucleus we have the DNA or chromosome present inside. So initially when the cell is not dividing, the chromosome is in what? It is spread, like it is in a thread-like form, that is called chromatin. Then when the cells start dividing, the chromatin convert into a condensed form, that is the chromosome. So here we can see the condensed form of chromosome. Then we have, so basically we have 23 pairs of chromosome in our body. Then the gene. Gene is basically a segment of DNA which codes for the protein. In our DNA, in our chromosome, we have many functional and non-functional part. The functional part we call it the exon, and the non-functional part we call it the intron. So, gene is what? It is a segment that is the functional. Functional means it codes for the protein. Then we have the other term, that is DNA. So DNA is the term that includes the bases. Bases means there are purines and pyrimidines that will talk about in the next slides. So I have a video to make you understand more clearly. So let's discuss about the Watson and Crick model of DNA. So here we can see the structure of DNA. It has two polynucleotide chains and it is double helix. Okay, so diameter of this is about 20 angstrom. It has minor groups and major groups. With 34 angstrom, one complete turn makes 34 angstroms, and the distance between two base pair is 3.4 angstrom. Okay, so let's discuss about the building blocks of DNA, that is the nucleotides. So nucleotides basically consist of sugar, phosphate group and nitrogenous base. The sugar is basically the backbone of DNA, and it is deoxyribose, which is a 5-carbon sugar molecule. Then we have the phosphate group, which is attached to the sugar. It gives the acidic character to the DNA. Then we have the nitrogenous base. There are four distinct bases like the adenine, guanine, cytosine and thymine, which store the information. Here we can see the structure of the building blocks of DNA, like the phosphate group, the sugar and nitrogenous base. Now let's discuss about the Erwin Chargaff rules, where Erwin's first Chargaff rule states that the base pair equivalence, where amount of adenine is always equal to the thymine and amount of guanine is always equal to cytosine. We should remember that adenine always pair with thymine by 2 hydrogen bond and guanine by 3 hydrogen bond. Then the second rule is the consistent base ratio across the species, where it states that the ratio of purine is always equal to the ratio of pyrimidine. That is A plus C is always equal to B plus C. right?
Corona.
Yeah, corona. So what was the genetic material in that coronavirus?
RNA.
RNA.
Yes.
Okay. So that is the RNA or ribonucleic acid, which you can see here. It is single stranded, and here deoxyribonucleic acid, which is double stranded. Other difference is the sugar. If you see the sugar of ribose sugar, here we have in the second carbon we have the OH group, but in deoxyribose we like oxygen. Okay, and the another difference is in deoxyribonucleic acid we have four bases, that is thymine, cytosine, adenine, and guanine. But in RNA we have uracil, cytosine, adenine, and guanine. So when here you can see instead of thymine we have the uracil. The next slide includes the difference between nucleoside and nucleotide. So nucleoside basically means the nucleoside basically consists of the sugar and nitrogenous base, whereas nucleotide consists of sugar, nitrogenous base, and phosphate group. Like here we can see, this is the base, then we have the sugar, and we have the phosphate group. Another video. Do the assessment. I'll have some questions you have to answer. Okay. First question is, what does DNA stand for? Option. के जाने चाहिए बोले को देवे देखो बोले मास्टर okay, the second question is, where is DNA located within the cell? Cytoplasm, nucleus, mitochondria, or cytoplasm? ये जाने चाहिए इप्सिता
Mitochondria is the
The options are straight line, spiral, circular, zigzag. ये जाने चाहिए को आपको
Top one till the second.
what are the building blocks of DNA called? Nucleotides, amino acids, sugars, phosphates. ये जाने चाहिए को
amino acid.
amino acids ये ले लो nucleotides very good. Next question is, which of the following nitrogenous base is not found in DNA? Adenine, cytosine, thymine, and uracil. अरे जोड़े को.
Uracil.
Next question is, how do the bases in DNA pair off? A with C, G with T. Good. David Rupo. Answer करो. Answer करो.